just my name is Ling, and I'm currently the co-founder and president for KMT. Within KMT, I'm responsible for the R&D and then product development. So my background is in biopharmaceutical industry. So my latest job is within NUS, and then and also as a principal investigator in a pharma company called B1 Medicine for cell and gene therapy. We start off this company based on the idea that we can actually integrate these AI platforms to solving the challenges in the biologics manufacturing. I will also have some polls that to discuss with the audience today as well while I run through my slices. So again, very happy to share my thoughts and share the views on what could be leading to the biologics manufacturing, particularly on ATMP onwards. So let's get it started. So maybe before we go into starting, I think there's a poll over here that to understand better about the audience, your backgrounds and which sections you are from, which divisions in there. So maybe you'll take a few minutes, take 1 minute to pause here to let you to answer the questions. And so, Christina, are we able to know the poll right away, or we will just wait until the last?
Yes, so maybe we give it another 10 seconds and then I will share the result.
Okay.
Okay. Can you see the results?
Yes, I can see that. So, well, thanks a lot for the engagement. So, and it's really nice to see that most of the people actually from regulatory sections and quite a bit from CMC. And from the other side, I don't know, most of people is actually more willing to know more about what's new about the AI, what's new about the biologics. Okay, so very very interesting audience. So what I mentioned the challenges in the biologics manufacturing. So unlike the other pharmaceuticals, like small molecules like Panadol or other things, those small molecules is actually manufactured through chemical synthesis. So which is relatively easy to control. And then we actually have studied this for many years. But biologics manufacturing is something different because biologics, for example, antibody, mRNA vaccines, CAR T cell therapy, even for cell as a therapy itself, is actually everything is produced by cell. So it's not. It's actually we are actually modulating something that we don't really understand pretty well, and to control then to manufacture the biologics. Unlike small molecules, like you can do a chemical synthesis like this way. So this is actually putting a lot of challenges for the biologics manufacturing. For example, in ATMP, we know that the CAR T cells, when you're trying to manufacture CAR T cells, there's a very high cost to do that. Part of it is because we still wanted to understand better about those cell therapy, how the cell is proliferations, how the cell is keep these phenotypes in the cultures. So one of the standalone is like the patients will probably need to pay about $500,000 for this therapeutics. This really limits the access to the patients. So we think about this way, and right now everyone is know that we're actually entering into a very exciting era, which is we have a new tools in science is called artificial intelligence. So that actually keep us thinking about how can we use AI to control the cell behaviors so that we can limit we can actually reduce the cost of making it. So in this case, when we reflect these ideas, we really think about that maybe we can do this way by twofold. One is to create a AI platforms, which is so-called virtual cells platforms, that which can digitalize the cells, and then we can discover the gene targets which can actually control the cell functions. And then as the next steps, we can design drugs to control the cells. So instead of designing a drug for human beings, we actually design drugs for a cell. So in this case, it's a two combination. One is from AI platforms that we can understand better about the cells. The other is we developed a tool that can really actually manipulate the cells, modulate cells in a way. So traditionally, there's some ways that already has been done in the field to control the cells. For example, media reformulations or cell line engineerings. So these are existing tools that we developed it to control the cells. They have their own merits. For example, media reformulation, we can actually manipulate up the nutrients compositions to better support the cell growth. But for the media reformulation, there's also has its limitation on there's no drug effect, meaning that we only provide the nutrients, but not really actually modulate or manipulate the cells to certain functions. So the other is pretty well known as like cell line engineering or gene editing. So you just directly knock out or enhance certain genes, and then you give the cells novel functions or control the cells in a way. However, the limits on this approach is not a turnkey solutions, meaning that once you actually done the engineering part on the cell, you really kind of cannot really turn them back. So a lack of on and off flexibility when you control the cells. And the bioprocessing or the even for the ATMP that you still require some cells to show certain traits at a certain stage. So you don't want it to have these certain traits throughout the whole process. So you need a ways to can quickly and on and off to modulate the cells. So we think there's a third way that it actually can do things that is actually design small molecule. It's pretty precisely control the genes once you add into this small molecule intermediate and it's taking effects, and once you wash away, and then it's losing off its effects. So it's really flexible and can tune the cells in a precise way. It has benefits for both patients and benefits for both for pharmas. For the patients, because we control the cell to proliferating better and then or producing antibody better, then we actually lower the cost of producing these biologics, then increase the access. Then, of course, the benefit for pharma is very obvious. If we can increase the efficiency of cost, then we actually have a better scale to run these productions. So in order to actually achieve this part, just mentioned that there are actually two parts. One is a virtual cells model. You need to find a way that it can digitalize the cells. The other way is once you identify the gene targets, then you need to quickly design the small molecule with the potent efficacies to against that genes. So this is the layout we think that we should actually put it up to build up these so-called cellular drug platforms or cellular drug generations platforms. So the first step is to find the targets, which is based on the virtual cells models. And this actually later I was talking about, well, I will show an example, run a case to see how the virtual cell platform works. So once you identify the genes based on these digital platforms, then you actually design a novel small molecule with high potent that can actually act on these genes. So then in the end, you can actually manipulate the cells in a way. But when you design these small molecules, unlike you design a molecule therapeutics for humans, then you need to consider PK/PD. In these biomanufacturing, when you design small molecules, you need to think about other dimensions. You need to think about how stable this small molecule can be existed in the manufacturing systems. So, and then also need to think about whether it can penetrate to the cell membrane to a particular cell type, either T cells, CHO cells, or HEK cells. Another part is that you need to think about the metabolisms, so on and so forth. So the thinkings behind designing a small molecule for the biomanufacturing is different with designing small molecule for human bodies. So to do this, it's really important to generate the data, which is pretty important for any kinds of AI model building. So since we have a human genome projects going on, there's actually a very abundant data existing out there that for you can to utilize. We have genomics, proteomics, we have protein structure data, metabolomics data on the cells, and anything else. The tricks or the important thing is how do you actually internalize this public or what we so-called auto layer data within your own use. So then this need to design a homogenized layer to homogenize the data so that it can use for your model. And more importantly is the inner layer data, which is your in-house data sets. This is how actually you differentiate your model with other people who actually utilize the out layer data, which is publicly existed. So in this case, a very important concept is called gene and cell perturbation database. It is like we need to dissect on what happened to a cell once you manipulate a particular genes. What is the transcriptomics look like, change look like, or what is the other proteomics change look like? So this is gene and cell perturbation database is actually very fundamental for building the virtual cell model. So right now we have more advanced tools. We can even zoom in to a single cell level to check what happens if just a single cells, when a particular gene is being knocked out or enhanced, are these omics data being changed? So this level of informations can really contribute to this virtual cells model building. For the model itself, then there's multiple parts that need to be considered because there are different types of data. There could be figures, that could be text, that could be also gene sequencing data. Different data source will need to have different models to pair with it. So then you can actually make sense, convert it as a sensible or calculatable inputs. So to do these cellular drug platforms, in the end, you have an output, which is which molecules or which gene targets that can really give the desired cell functions. And then because you pair with the chemical design small molecules, then you in the end will get another output is the small molecule with high potent efficacy against that gene targets. So I'm going to run through examples to showcase how this system can work, so to better understand the parts. So we wanted to solve a problem is very important in the T cells ATMP domain is how can we actually let the T cell expand well at the same time to let them not lose the memory or enhance the exhaustion features. Because sometimes what we know is if we want to let the T cell proliferating faster, sometimes actually most of the times they will lose the immunophenotype, meaning they will reduce the memory and then show the exhaustions more. So we really wanted to see what is the other ways that can balance both quantity and quality. So this is as an input put into this virtual cell model, and then the system starting to working on based on the data I just described. So this is step one, that to find the gene targets. After we find the gene target, and then we do de novo generate small molecules. This compared with screening is much more cost efficient, and right now we have a better tool to do a precise de novo generations. Okay, I think there's also a poll over here. Is that the case? Yeah. So this is also interesting to wanted to understand better about when you actually wanted to design for example, a T cell pipelines, and when you wanted to manufacture it, and how do you actually optimize the whole process? So I would take it for be 1 minute for you to write it on. that. Okay. Okay, Christina, so do we have the result already yet? Okay, that's great. So yeah, I can see most of the people writing over here is done by experimental try and error methods. Um, and I can see that the next one is digital models and advanced analyticals. So yeah, from the poll, I can also see that actually currently, even right now, under our this uh, era, and we have working on computer for so many years. Still, most of the people or most of the time, we are still relying on experimental try and error. Okay, so then back to the parts. How do we actually can really eliminate the experimental try and error methods? This is what we think as the nowadays with the AI tools become more and more robust. We are actually able to do this, just like when we use the AMS to solve the protein crystallization methods, the problems. So just now I highlight into several data structures. So firstly is the T cell omics data, which is the T cells when they at the quiescent status, how a T cells look like in general in different angles. So in this case, we collect single cell sequencing data, transcriptomics data, proteomics data, and epigenomic data. Basically, this is to lay out how the T cell look like at the beginning as a day one. And later on, when just I mentioned at the beginning, the gene cell and gene perturbation is pretty important. So then, in this case, we have checking on several function for T cell, which is very important. Proliferations, interferon gamma, TNF, L2, persistence, low and high, and then we do these gene perturbations on every single assets, and then collect these gene perturbation data sets as well. So overall, these data sets, when we collect it, we will put into the algorithms. This will be actually, in a way, the whole background of the model is a bit complicated, but in the end, in a nutshell, is to let the AI to learn to zoom into the cellular level to see how a cell will react once a particular gene being manipulated. And then the AI right now can be actually understanding how a T cell works better than us. Um, so then after they virtualize the T cells, and then we just input our query, just tell me how actually I can find a gene targets that can actually balance the quality and quantity. So the system actually give back about several candidates, and it will run through one by one on the scenarios, and then we actually just test that, and then ultimately design small molecules that we can test in the wet labs. So so this is actually how the system right now can actually run through the simulations. So assuming that you have a gene A, and then you wanted to know what happens if I knock out a gene A to the T cells, and then based on those data structures and based on those things that he has been studying, and then it can tells you that hey, this is expansions how it should look like. This is actually purely simulated by the model, and how its memory should look like after the gene A has been manipulated or inhibited. The central memory will increase, stem cell memory increase by how much? It will actually very accurately or very precisely tells you a number. And I put it here as a true data, which is actually later on when we do the downstream wet lab validations. And this is the actual wet lab data we collected. And you can see that from the model simulation to the true data comparisons, they're actually reflecting to each other quite well. So that proves the robustness in a way or accuracy in a way these virtual cells can can be done. So the data input is very important in this case. So the system can right now just run through these genes one by one, because in the end we only have about 20 to 30,000 genes. That is, we can do it. So just quickly run. It can actually run every single genes, or you can just let it run to a certain point that suffice you to do experiments. For example, in this case, we just let it to run suffice the genes numbers good enough for us to do the downstream validations. So we shortly several one because all actually human brain is designed not to really analyze the high dimensional data. The most convenient is just tell me one data, whether it is yes or no. So the system actually can actually combine all these features and then put it at one likelihood score. So we put a cutoff, for example, 0.6, and whatever is above 0.6, then we will shortlist it and for the downstream validations. So this is step one, that we collect the data, we put it into the machines, put in the AI to learn it, and then let it to simulate, help us to find the targets. Okay, so the step two is once we identify the gene targets, then we got to design the small molecules. So in this case, there are a lot of models out there that are able to use it. For example, BOS2 or Chai Discovery. There's a lot of small molecule design tools that is available currently. But you have to also to keep in mind this is in the biomanufacturing field. So then you have to also tweaks the molecule design based on the biomanufacturing scenario. You have to make sure that the small molecule is dissolvable, that you are not making the pill, you have to dissolve in the culture, you have to consider about the cellular metabolism for these small molecules. So then I will just cut you off on what's the details, how to design the small molecules. And then in the end, we generate, based on the targets, we generate one small molecule for one target and to do the downstream lab validations. And this is how the data look like. So in the end, we just get 11 gene candidates. So we synthesize 11 small molecules for each of the gene targets. So each one number 1 to number 11 representing different gene targets, different small molecules. So basically you need to do several runs, run 1, run 2, and run 3. So in the end, you screen out those things is not really promising and leave out the things that is actually repeatedly shows a good results. So in the end, we got 5 small molecule and 5 gene targets for another level of lab validations. So from here, you can see compare with the control, which is we don't add this the small molecule compounds. You can see that all of them actually quite robustly increased the expansion of the T cells. That this will be 5 to 10 folds higher of the T cell expansions. The next question is, other than expansions, what about the rest, like immunophenotype, or about its therapeutic efficacies? So for this, out of these 5 molecules, you can see there are several molecules is really standing out. For example, number 9 over here. For the others, you can see very interesting is that it can actually also keep the memory retained or enhanced, for example, 7 and 10. So that actually also reflecting we do have a way that it can actually increase the proliferation at the same time keep the quality. But I guess they are actually acting on different mechanisms. So we think compound 9 is pretty promising. So then we just use that for the other level of wet lab validations. So we also check the exhaustions. So you can see after adding the compound 9 that the exhaustion level also being decreased, and you can see that increase of the in vitro activity is being enhanced. Most importantly is the in vivo efficacy, which is very important and gold standard test you have to gone through by doing CAR T therapy. So this is a CD19 CAR T, which just prove a concept to demonstrate that it actually, based on these features, it does have improved the in vivo efficacies. So you can see in this CD19 CAR T has a very good control on the leukemia in a mouse model. The mouse uh, that receive a CAR T, which is culture with the presence of compound 9, can increase its survivability significantly versus the one that without culturing through compound 9. And this is actually before injecting to the mice, the compound 9 is being washed away. So it's kind of a way to modulate the cells to become a better status before we put into as a therapeutics into the mouse. So this is an example how we actually leverage the whole AI platforms and design small molecules to control the T cells to be proliferating faster and then push them to get at a better immunophenotypes. So we got to know what is actually the mechanism behind this, right? So from when we run through these AI systems, AI platforms, that already tells you what is actually the directions for these mechanisms. So it points out actually addressing the T cell senescence will be a key to balance the expansion and quality. This is actually produce a really blind spot for T cell manufacturing because people wanted to actually more focus on exhaustions or different cytokines combinations to modulate the cell's memory. But there's a seldom solution for addressing T cell senescence, which is pretty important for T cells therapy. So the compound 9 is actually just to modulate the activated T cells to be less senescent, younger, more robust T cell status that will reduce exhaustion, maintain the memory, and increase expansions. So this is some other wet lab data to show that you can see a surface marker CD57. So when you add in the compound 9, you can see that the CD69 is pretty low in a way. So meaning that it is working on the senescence, reducing the senescence. You can see that CD57 is keep increasing without in the normal cell cultures. But when you add in the compound 9, kind of suppress that the cells going into the senescent status. You can see all those senescence driver genes, CDKN2B, N2A, and P21. So once you add the compound 9, the T cell become less senescence. Those driver genes being heavily reduced expressions. And not just by those single genes check, we you still need to check the overall landscape landscape of genes signatures for the T cells. So in this case, we culture checking the whole trajectory in the T cell culture, day 5, day 10, day 15. So in the control groups, which is we don't add the molecule to tilt the cells to a certain state, you can see from day 5, the gene expression is like this. And then gene 10, some of the genes gradually progressions to these sectors. And on day 15, that most of the genes is overexpressed by this part. So further analysis on this part is reviewing that this is actually quite a lot of with T cell quiescence or apoptosis over here. So meaning it's like a late stage of T cell culture status. But when you're adding the compound 9, at the first beginning, you can already see, because it's a drug, it's taking effects very quickly. It infiltrates to the cells and act on the genes very, very quickly. So at day 5, you already see that gene signature is quite different between with compound 9 or without compound 9. And on day 10, you can see that now the gene is actually quite expressed in this late stage T cell gene signature. Most of the gene signature is actually expressing in here. This is actually more associated with T cell growth and protein synthesis, meaning the T cell is actually at a very robust stage. So through here, you can clearly see that actually adding the compounds to modulate T cells you can kind of delay the T cell entering into later stage of T cells. So that actually demonstrate the purpose. So T cell senescence is very important. It's unlike exhaustions. Um, Senescence cells has also been shown that will reduce that the T cell or immunotherapy efficacies. You can see that in the gastric cancers, this CD57 low T cells patient cohort, they have a better survival versus the one who actually have a high CD57 T cells. And then people do these experiments also testing out those, they use irradiation to induce the immune senescence. You can check out that if the immune senescence is being induced, then you add in the PD-1, that actually the cells will actually will have poor tumor control on the tumor progressions. Vice versa, if you eliminate the senescent cells, then when you use the immuno checkpoint blockade, the tumor is better controlled. Senescent cells gradually, you can see just as show that more CD57 will become more and more. So one way is you can also see that actually those senescent T cells will be has less transduced by the viral vectors in this case. So in this case, that also explains in a way why senescent T cells, well, if you have a higher population of senescent T cell, the efficacy of the immunocell therapy will be lower. So once you address the immunosenescence, then what would be the other benefits other than keeping the T cells expand well at the same time it keeps its immunophenotype? Well, in a way, it's actually keep the T cell more robust. So because T cell is very personalized, so some of the pool donor, their T cells wouldn't grow pretty significant. So also they can't really actually receive CAR T cell therapy in the end. So then in this case, once we address the senescence by adding the small molecule to modulate the cells, we can see the poor donor right now, this show as orange. Right now they can actually grow exactly the same as a good donor. And then if the good donor adding the small molecule compound 9, that is just expanding much better as well. So and then the trick is really to see the viability. So when this poor donor grow into the culture, they just keep dying in the culture. And when you add in the compounds 9, which is addressing its senescence, the T cell become more robust. So you can see that this poor donor is right now showing that as an orange here, they are basically has very high viability versus without adding the compound 9. So another part is that since the senescence is really, we know like senescence is working like pressing the brake for a car. So then in this case, do we really need to press a lot of gas, press harder on gas? So I think the answer is not really. So based on this hypothesis, then we're trying to see if we can reduce the cytokine use, because cytokine is actually acting like pressing the gas. So in here that we gradually reduce from 300 unit IL-2 all the way down to 10 unit of IL-2. Maybe we could look at the right figure first. So this is a control group with 300 unit IL-2. So they grow as a blue curve over here. And with the compound 9 to adjusting its senescence, then we gradually from 300 unit all the way down to 10. You can see at 100 unit of IL-2 that actually grow similar amount of the T cells compared with 300 unit IL-2, but without compound 9 modulations. So it does mean that you can have 3 times reduce of cytokines, which is actually reduce 3 times of cytokine cost. So this is one thing. The other thing is you can see that the T cells status right now actually also become better. So the CD57 expression level is much lower than the control groups. So in here that you have a better cost control. Also, you have a better cell status, so which is actually very good ways to manufacture T cells. And once you also addressing the T cell senescence, T cell become more robust. Right now, most of people is in the field is trying to automate the T cell manufacturing process. The automated machines comes with several problems as well. So when you wanted to control those critical process parameters, for example, temperatures or CO2, or to maintain the pH values, sometimes the machine can be faulty, meaning that they can't really actually control this temperature within the certain range. They will lose the control. So we simulate the extreme cases that we stop the CO2 injection, we stop the temperature control. You can see this is stress. So when the stress is coming out, the T cells already suffer, and then this is the parts cannot be reversed. So when we switch on this temperature control or CO2 control after 12 hours, when the cell become apoptotic or this program is being initiated, they were never able to coming back. But when you add in small molecules, adding there to maintain the cell use level or reduce the senescence, you can see in the end the cells will gradually can coming back to that part. So it means it kind of like give the cells a safeguard, so you don't have to worry too much about that the machine get faulty on this. The overall T cells become more and more robust, also being demonstrated by like when you do the freeze and thaw cycles, which is also important for the T cell therapy. We know that when the T cell or particular for some donors, when they freeze down, delivered to the hospital before they get into the patient into thaw back, get recover. So sometimes or some of the donors, or most of the cases, actually the T cells or lymphocytes efficacy will reduce. So we check on this. What happens after we actually modulate the senescence? So we compare with before freezing and after freezing. So before freezing, if you're addressing the senescence, you can see this red bar versus the blue one. That already increased the cell's robustness or cell activity. When you actually recover the cell after thawing, first thing you can see actually, for example, we'll compare this to this. You can see like it's a bit of reduction of the T cell activity by secreting interferon gamma. And some of the donor, for example, donor 2, which is quite being affected by this freeze and thaw cycle. So this is how it looks like before freezing as a fresh samples, while you're adding the compounds that increase that is activities. When you actually thaw the cells, you can see this blue bar right now. They are heavily being damaged. Only leave about 20 to 30% of the cell viable cells or the activities. But when the cells addressing the senescence, then most of them actually preserve their activities. You can see they are still at a very high capability to secrete interferon gamma. So that indicating that if you're addressing the senescence, the T cell become more robust to counter these freeze and thaw cycles. And another part need to highlight that in the biomanufacturing, then designing small molecules to modulate the cells is pretty promising. But another part is also need to think about the regulations and think about how those regulators will think about adding the small molecules. So the good things about designing a small molecule by this AI system is that you can put into a lot of thinking or embedding about how you design the small molecules. So we design these small molecules as a pharmaceutical level or pharmaceutical standards, meaning actually this small molecule, when they designed, that already think about this off-target effects or its safety profiles. So, to demonstrate this, the compound 9 is being directly injected into the mouse for in vivo monitorings. So you can see that the high dose of plasma concentration has been detected because we inject this compound 9 for throughout the 10 days, inject every days. And you can see it's actually 200-fold higher than the amount we cultured the T cells. And there's no mouse weight change or there's no behavior change that demonstrate that actually the compound designed through these platforms can be very safe and to the pharmaceutical standards. This actually can also pave the way for combination use in the future, just directly to let the patient consume this small molecule and we modulate the CAR T cells in vivo. So looking forward to forward looking about these platforms. So small molecule is acting like a tool to manipulate the cells. And then the AI platforms is like building a virtual cells that can simulate the cell performance and behaviors. Um, Actually, these AI platforms can actually further be used as an intelligence layer for people to run the process. So traditionally, just I mentioned that people wanted to do troubleshooting or to change the design of experiments by try and error methods. Usually takes try this and that, and that will actually is time consuming and cost a lot of energies. And the another part is there's no like a real-time feedbacks. There's you don't know about the cell status in between. So it's really like a black box against guessing games. But if you have these virtual cells AI models, um, you can just simply put into your hypothesis, like you have this DOE, and then let the system to tell you what will happen with the T cells, what will happen with all kinds of cells, whether they will survive good, whether they'll produce high titer antibodies, so on and so forth. Or you can actually just put in a simple query, questions to troubleshoot to find the root cause. What actually caused the failure of this batch, and then how can I learn from my previous experiments or previous experience converted to a success for my current batch. You just simply dump in, for example, your past Excel sheets, your past PDF reports, and then can help you to process it. And so the output, it was tells you very accurate and real-time monitoring on the cell status, and or just facilitate to tell you what will be the critical process parameter, what is the CQAs, what will be the PQQs that for to help you to make the decision makings. So I think for looking really well excited to see more how AI actually can play a critical role for the future biologics manufacturing. Yeah, so with that, I want to end my talks and leave more time to further discussions. I believe also there is a poll here for the audience in the end. Okay, I'm also happy to take any questions. So Krishna, how should we do the Q&A? We just see the chats or
So thank you, Ling, for your presentation, which is very interesting. And also you might want to comment on the poll in a minute. While people are answering, please do feel free, everyone, to either put your questions in the chat for Ling or unmute yourself and ask him directly. So you've got 10 minutes in which this is a golden opportunity for you to ask anything about this presentation or anything related. Yeah, make the best use of this time.
yeah. I also see the poll results. Looks like there's quite a high number of people interested to try out the AI system in the future, which is very happy to see that my talks is effective to convince people.
Yeah, looks like pretty much everyone's interested. somewhat interested. So yeah, that's great. Thank you for that. And we also want to thank Ling for staying up very late for us. I think it's what, is it 1 o'clock, 2 o'clock over there for you? He's in the West Coast right now, aren't you?
Yes, it's probably yeah.
So thank
Two,
you
two.
so much for taking the time
No
out
worries.
to share with us this afternoon.
There's a question in the chat. Actually, Gary has a question, Ling. What's the objective with discovery of these new small molecules, and are you creating a product?
Okay, yes. So thanks for Gary for your question. So the objective of discovery those new small molecules is really to give a way or tools for us that able to modulate the cells. It's kind of like a hammer or like a tweezer that you can actually just poke the cells to let them to act as a way that they desired. And when you say, are we creating a product? If you refer to the small molecule, yes. If we within our company, that we will convert it as a product and become off the shelf, so everyone can use it. But in the future, our vision is everyone can be a customary ways. So, for example, you're just sitting there and you want to control your cells, and you just queuing your query, and then there's a small molecule designed, and with all the considerations, comply with the regulations, and then next day you have the small molecule at your door, and then you can use it to manipulate your manufacturing. This is actually the visions we are looking for in the future.
Ling, there's a follow-up question from Gary.
Different phenotype. Yes, that's a very good question. So just at the beginning when I mentioned about the virtual cells, so it can actually act on very quickly. So you just need to give the patients initial informations, like just give a transcriptomics, like mRNA sequencing, and then put that sequencing results into the models, because the model has already know how the T cells will react. So it will give you the information, some informations to know how to deal with these patients, what kind of small molecule is needed to achieve your purpose.
Fantastic, Ling. So that's very insightful. One question that I have in mind, I'm not an AI expert, but you mentioned a few layers, right? So you need to have multi-omics data, and then the gene cell perturbation, the different wet lab data. So how much data is needed? You know, you are also alluding to large language model. So it sounds quite daunting, to be honest. So tell us about how much wet lab data is needed, and where do we get hold of this AI model to make sense?
Yeah, that's thanks for the question, Pei. I think this is really a good question. Like how much is needed, or how much is sufficient. I think we always looking for these answers. I think gradually we will know, in the end, maybe have a sense on how much is that. At the first beginning, when we do the explorations, is just like the more the better. Just create it, whatever we can get, exhaust all the resources to get these things, to build this virtual cell model. But because right now we also do gene labeling, we also totally draft a new kind of how the gene is interactions within the cells, to get a maps on a particular cell types. So I guess throughout this activity, maybe we virtualize several cells afterwards. The system or the model itself will better and better understand how a cell signaling pathways react. So the requirement for the data input will be less and less. So that's what I can feel. But in terms of exactly how much, for for now, I don't really have the good answer for this. From the current activity on the T cells and CHO cells, we usually definitely have to be several billion or 10 plus billion of sequencing reads to finish these digital cells constructions.
Thank you.
Does anyone else have a question? Otherwise, we will let Ling get some sleep and bring this session to a close. We'd like to thank you very much for sharing your expertise with us, and for our audience for joining us this afternoon. We have recorded this session and it will be saved into the ISPE Singapore YouTube channel, so that you can access that if you missed any of it or if you'd like to re-watch. So unless anyone else has a question, we will bring this session to a close and thank everyone for joining our first ATMP COP session. So thank you, everyone, and have a lovely evening and a good rest
Thanks,
for you,
Ling.
Ling.
Thank you, and
Thank
hope all
you.
the sessions going very successful in the end.
Thank
Thank you very
you.
much. Yeah.
Thank you.
We look
everyone.
forward to seeing you. Thank you. Bye-bye.
Bye bye.